Hey guys, I'm Harsha and I brought a new lecture today with me regarding the leukocytes and the production of antibodies. The production of antibodies might be a nightmare for most of you, I know. And this lecture should seem like a bedtime story that will convert your nightmare into a happy dream. So let's get started. Have you ever got injured? Of course, you must have. Have you ever thought after you are having the wound on your body, a cut on your skin, what might be the after effects? The pathogens, the bacteria, they might be crossing the epithelial limit and entering your connective tissue. And once they come to the connective tissue, they start invading the living cells of your body. By that time, many events take place inside the connective tissue from our side also. The neutrophils wandering nearby in the microcirculatory system, the monocytes and the dendritic cells, they start their phagocytic action. First, the neutrophils, which are most abundant leukocytes in the blood, they come out of the capillaries by diapedesis, by the help of their pseudopodia, and try to phagocytose, try to eat most of the bacteria or pathogens as maximum as possible. But still, there is not much attraction of the neutrophils. The neutrophils are not able to know where are the bacteria, which cells have they invaded. So that's why the process is very slow. But there needs to be some sort of conspiracy, some sort of programming, planning that will render the neutrophils to get attracted towards the pathogens, bacteria. And this will be the main focus of the story today. Now, after neutrophils, the next prioritized cell is monocyte. Monocyte also come out of the blood vessels, the capillaries, by diapedesis, and they are also phagocytic in nature. They also try to engulf most of the bacteria. But once they come out of the bloodstream, when they wander in the connective tissue, they are no more called as monocytes, they are called as the macrophages. And macrophages and the similar cells like dendritic cells, Langerhans cells, they are also called as the professional antigen presenting cells because they are going to present the antigens to somebody. Now, who is that somebody? That's the main question to be answered. But before that, let us be clear with the summary of the video and then we shall proceed. You might be knowing what antigens are. Antigens are any alien molecule, any foreign molecule that has entered the body and that is not recognized by our self cells. Our self cells are going to fight against it somehow. Now, those molecules are called as antigens. First, in this video, we shall see how an antigen that is produced by the product of digestion of the bacteria by the neutrophils and macrophages. Of course, the antigens are produced. How these antigens are processed? Apart from digestion, they need to be processed. They are processed inside the APC for presentation. Just like a gift needs to be wrapped before it is presented. Or you may say a gift needs to be handed over properly. Now, to whom the antigen will be presented? That's a very big question that needs to be answered properly. You need to wait for that. And the third, what will be the aftermaths after presentation of the antigen? To whom it should be presented. Now, in the third number, you might be knowing, you might have come to know that the present is going to be given to the lymphocytes. Now, which lymphocytes? That is still a question. Next, we shall be coming across different MHC, CD, IL, ILR, and the genes that produce different IL and ILR. Next, as per the serial number 5, we shall be knowing about the process of antibody formation in B lymphocytes. Actually, when the B lymphocytes or its descendant start producing antibodies in huge number, they are called as plasma cells. But these need to be stimulated somehow and helped by the T helper cells, one type of lymphocyte, of course. Next, we shall also know about the story of cytotoxic T-cells, how they function, how they act upon the antigens or the target cells. What target cells have they got? Okay. One thing I forgot. According to the serial number 4, there are many abbreviations here. MHC, CD, IL, ILR, genes for different IL and ILR. So A, B, C, D needs to be clear, I think. MHC are major histocompatibility complex. In humans, they are also called as HLA, of course, and they are produced by the macrophages. They are numbered with Roman numerals. And what is the criteria of their numbering? In this video, we will come to know. Second, CD. Cluster differentiation complexes are found over the surface of leukocytes, many leukocytes. And according to their molecular structure, they have been also named by numbers. The first discovered CD has been named as CD1, next CD2, and so on, according to chronology. Third is a very interesting thing. IL interleukins are just like hormones. They are communicating molecules between the leukocytes. Inter means between, leukins refers to between leukocytes. What are ILR? Of course, as the name suggests, these are like the golf pots or golf holes inside which the golf ball fits in. Interleukin 1, suppose IL-1 will fit into IL-1R. Interleukin 3 will fit into IL-3R. Interleukin 4 to IL-4R. Similarly, we can also say that interleukin 1 will not fit into IL-3R or IL-4R. It means they are very specific in their recognition. IL receptors are also found on the surface of the leukocytes. One leukocyte sends or secretes the IL and another leukocyte catches that IL by the ILR present on its surface. Only then the communication is established. We shall also come to know about the genes and the chromosomes in which they are present for different IL and ILR in this video. So let's get started. So we shall start from the neutrophils first, because it is easy to understand the action of neutrophils, and then we shall go to other leukocytes. Now let's see what the neutrophils does. Already we have discussed, if an injury happens and the pathogen or bacteria has entered, first of all the encountering cell are neutrophils. Now neutrophils will try their level best to phagocytose maximum of the pathogens. But by this time the neutrophils are not getting attracted sufficiently to the spot of injury. That's why there must be some event, there must be some conspiracy for attracting the neutrophil to the injury spot. Now we will see what conspiracy has to take place. That's a different thing. But first of all, let's see whichever pathogens come in front of the neutrophil, how does it act with them. First of all, they will form a food vacuole just like the protozoans. Inside the food vacuole, the pathogen or the bacteria shall be digested, and the products of digestion are nothing but the antigens. Now these antigens are released outside as residual products, and then next is these antigens will be attached to the pre-programmed B lymphocytes on their surface. Now see, now let me tell you a story about the B lymphocytes. Every B lymphocyte has many surface antibodies attached, and every B lymphocyte is pre-programmed to attach a certain antigen only. For example, see here I have drawn two B lymphocytes. One B lymphocyte is pre-programmed to attach the circular antigen, and another B lymphocyte I have considered here is pre-programmed to attach the triangular antigen. See here. Now this B lymphocyte is attaching only triangular antigens on its surface. Triangular in the sense I am taking a certain antigen, a peptide or a glycoprotein, an antigen that can fit here. Now it may not be triangular. For convenience I am taking the shapes. Now similarly another B lymphocyte, it is attaching only the circular antigens on its surface. Okay. Now for a while let's forget this neutrophil, this B lymphocyte, everything, and also the antigens, and let's study the story of the APC first, and later on we will come back to this story again, and we will try to merge, connect both the stories and create a new story. Let's see what is the story of the APC. Now here I have taken a macrophage as the APC. Now dendritic cells have philopodia on their surface. Otherwise they are similar to the macrophages only. So I have taken a macrophage here. Now see, the macrophage first of all encounters with a bacteria, and it is engulfed a bacteria inside a food vacuole, and then it is trying to digest it, and after digestion the antigens are absorbed into the cytoplasm of the macrophage, and then This activity, this event stimulates certain genes inside the nucleus to produce a certain molecule. Actually, the stimulation is done to produce certain molecules called MHC. We have discussed it already about this MHC and what is the speciality in the MHC? These are produced by special genes called as suffling genes. They are present in the sixth pair of chromosome, and the suffling genes produce these polypeptides, the MHC, in a very peculiar manner. The genes continuously shuffle and produce varieties of MHC. Every MHC is very special in its structure, but there is a constant portion in every MHC and there is a variable portion also in every MHC. Now see, I have taken the shape of the variable portions in a very intentional manner, that one is semicircular, one is fitting with the square, one can fit with the star, one can fit with the triangle, and one may not fit with any body. Now these antigens will try to fit on these MHC molecules. Now see, the MHC molecules can be categorized into MHC1 and MHC2. I will tell you the reason later why they are categorized as MHC1 and MHC2 shortly. Within few seconds you will be coming to know why they have been categorized like this. But for the time being, let's say the MHC1 has the circular antigens to be fitted upon and the square antigens to be fitted upon, while the MHC2 have the star antigens to be fitted upon and the triangular antigens to be fitted upon. Now after the MHC are produced in such a manner from the suffling genes, they will travel to the surface. But one thing to remember, the most interesting part of this story is that every MHC will take an antigen on its surface and then it will appear on the surface, as if we are handing over some flowers to a friend. Means it is taking the gift. The antigen can be considered as the gift, and the MHC molecule can be considered as the hand which is presenting the gift. See, you cannot just leave the gift in front of a friend without handing it over. You have to hand it over. You have to hand over the gift to somebody. Similarly, like handing over the flowers to somebody. That's why the MHC are molecules that can be compared with the hands that are presenting the antigens, and that's why the name is professional APC, because this cell knows how to present the antigen. Now let's come to know to whom it is being presented. Now all these events have taken place to present the antigen to somebody. Now who is that? Who is the great person to whom the antigen is being presented? Now so some antigens here have been selected and along with the MHC they have been presented, see, but some MHC could not be selected. Why? Because it couldn't get the particular antigen it is meant for. Probably that antigen it is meant for is not being produced from the bacterial digestion. So now next thing is to whom it is being presented. Now there may be two entities, two leukocytes to whom it may be presented. Specifically speaking, they are not only leukocytes, they are lymphocytes. Now who are they? The first one we will try to explain is our T helper type 2 cell. Now type 1 is not relevant here. Type 1 is present in the bone marrow, that's why Now we shall be considering only the type 2 cells here, T helper type 2 cells. Now these cells also have another name, CD4 plus. Now why they can be called as CD4 naive cells? They are called naive because they are still not active. Now they will become active when they receive the gift. Now what is the gift for them? The gift for them is the antigen. Now how will this T helper cell receive, and on what criteria will it receive the gift? See, every T helper cell has CD4, no doubt, but along with that it is also having some TCR. What is TCR? TCR means T cell receptor. Now every T helper cell has a particular type of T cell receptor only, and that can fit with a particular antigen only. For example, this cell is having the TCR that can fit with the triangle antigen only. Now the helper cell observes that already the gift is being presented in a very specific manner. Along with that, the MHC2 is um, giving the gift, and the MHC2 is recognized by the CD4. The antigen, the gift is received by the TCR. Now see, here I would like to say why this is called MHC2. That MHC which can be recognized by the CD4 is called as MHC2. MHC2 is actually related to our T helper type 2 cells. Okay, now both the things have been done. The gift has been presented, it has got attached to the TCR, and the MHC2 also has been recognized by the CD4. What next? Next is a communication. This is just handing over the gift. That has been done. But the presenter must communicate with the helper cell also. Now that's why here the macrophage is saying I need to talk also. Otherwise, probably you will not understand why I am handing over it. It appears as if it is trying to speak to the helper cell. Now at cellular level, we all know that the language is like cytokines, hormones, etc. Here it is interleukin 1. The first interleukin, for the first time we are coming across an interleukin, and that's why we are naming it as IL-1, interleukin 1. Now interleukin 1 is released or secreted by the macrophage, the APC, and it is received by the T helper type 2 cells. And what is next? Now both the cells are shaking hands with each other, like these two people. They are shaking hands with each other. It appears they are one is giving the gift, one is receiving the gift, and they are communicating with each other. So there is a bonding, but for some time only. Afterwards, what is the aftermath? We shall see later in the next slides. Now let's forget this story also, the T helper type 2 cell story. Let's come to understand a first a plus, that is cytotoxic T-cells. Now see, suppose the macrophage comes in contact with a CD8 plus cell. So the CD8 that is cytotoxic T-cells, if CD8 complex is found over whole surface of the T lymphocyte, it is called as CD8 plus or cytotoxic T cells. But since they are inactive, they can be called as naive cell. So this cell was a CD8 plus naive cell till now. But after it comes in contact with the MHC1, so it means that this is also presenting the gift, the antigen. But MHC1 is recognized by CD8 and the antigen that fits with the TCR present on its surface. See, one lymphocyte possesses only one type of TCR. Now, if this TCR is fitting with the antigen here, then coincidentally both of them are fitting each other. What? The TCR is fitting with the antigen and the MHC is fitting with the CD8. So this communication is also over. But what next? Now this is a very short story. Let us discuss it immediately. Now these two cells, the CD8 plus and the macrophage, they are shaking hands like this. They are communicating with each other. And the CD8 plus cell is no more a naive cell now. Now it is activated. It was inactive, now it is activated. Now what next? Now it becomes cytotoxic in reality. Now what do you mean by cytotoxic cell? Cytotoxic T-cells are destined to kill cancer cells, damaged cells, virus-infected cells. And how? By releasing cytotoxins like perforin, granzymes and granulysin are injected, and perforin perforates the target cell. Granzymes and granulysin enter the cytoplasm and trigger a new pathway, the cascade pathway or reactions that lead to apoptosis. Now, what is apoptosis? Apoptosis is programmed cell death, and every cell has this program inside it until this program is activated. Now, granzymes and granulysin activate this program inside a cell. So that's all about cytotoxic T cells. Let's come back to our stories, old stories that we have left behind. Now let's come to those stories again. Remembering that shortly we discussed about the neutrophils, the antigens produced from them by digestion and exocytosis of the residual matter, and how these antigens fitted upon certain B lymphocytes like this. Now the story continues like this. We are having some B lymphocytes possessing the triangle antigen on their surface. We are having some T helper type 2 cells possessing the triangle antigen on its surface. So both of them are related now. So both are now called as clonally selected lymphocytes. And this process, this phenomena is called as clonal selection. They have now been clonally selected. For proceeding further, other lymphocytes, they may not proceed further because they may not be having similar privileges. That another T helper cell is also with the same circular antigen, it may not happen. But if it happens, that also will occur simultaneously. So let's take the example of the triangle antigen. The T helper type 2 cell is having the triangle antigen on its surface. The B lymphocyte is also having the triangle antigen on its surface attached to these antibodies. I forgot to tell you that these antibodies are always of IgM or IgD class. Remember, these surface antibodies of B lymphocytes are always of the group of the class IgM or IgD. Now these two clonally selected lymphocytes, what are they going to do further? Let's see. But let's summarize what we have learnt till now. We have not learnt much, but it is necessary to summarize that thing because we are going to learn the most complicated thing in near future. So first of all, let's summarize that. Till now we have completed the stories of neutrophils. Yes, how neutrophils phagocytose the bacteria. Second, how the macrophage and other APCs are successful in presenting the antigen. And thirdly, we have learned the story slightly about T helper type 2, but not thoroughly completely. And we have completed the story of cytotoxic T lymphocytes. But further, our main focus will be on the stories of chosen, that is clonally selected B lymphocyte and its next generation clone cells. Means the chosen B lymphocyte will undergo mitosis and the result daughter cells will be called as next gen clone cells hereforth. Similarly, the chosen or clonally selected T helper type 2 cell will divide rapidly and produce daughter cells which will be called as next gen clone cells of the T helper type 2 cell. Now let's start our story from a B lymphocyte. That B lymphocyte which has been clonally selected. What happens to it? Now we have taken that B lymphocyte which has the IgM or IgD antibodies on its surface which have got attached to triangle antigens. Now what is the next step? The next step is that at the base of the antibody CD79 cluster differentiation 79, probably these were discovered very late, that's why the name 79, the number is 79. They are present at the base of the antibody which stimulates the nucleus, somehow the genes of chromosome 16 to produce a certain IL receptor, that is IL-4 receptor. Now what is IL-4 receptor? Let's see why this is produced. Obviously I have already told that IL-4R will fit into IL-4. That's why this is being produced. Now who will produce IL-4? That's a question. Next thing is obviously the receptors will travel to the surface and on the surface they are waiting for the IL-4 to come. Now who will produce the IL-4? IL-4 will be produced by the clonally selected T helper type 2 cell. This IL-4 will fit into the IL-4R. In the meantime, you can also see that the same cell is also producing another interleukin that is recognized by itself and that causes autostimulation for rapid mitosis. So this clonally selected T helper type 2 cell is autostimulated. It stimulates itself to undergo rapid mitosis and form the clone cells, next gen clone cells. Now it appears that as if the IL-4 is like a golf ball and it is going to the pot of the golf and fitting into the IL-4R. IL-4R is just like the pot of the golf ball. Now it seems that both the cells are communicating. That's why the name IL, interleukin, is the mode of communication. I told you the cells are communicating. It appears that two memory devices are communicating with each other. Okay, now let's move forward. After this event happens, after IL-4 gets attached to IL-4R, it stimulates the B lymphocyte for rapid mitosis. Now this story is about the formation of the clones. Now see, the clones have been formed. This clone formation by B lymphocyte can be regarded as the clonal expansion or clonal proliferation giving rise to next gen B lymphocytes. Okay. Now these will in near future produce the antibodies, but before that there is aftermath. What happens? Like the B lymphocytes, we have already seen that the T lymphocyte also get clonal expansion. The T helper type 2 cells also proliferate like this by autostimulation, obviously. Next thing is the clone cells, B lymphocytes clone cells and T lymphocyte helper type 2 clone cells, they will try to communicate with each other. Now let's start from the B lymphocyte clones. First of all, they have to prepare themselves in order to communicate with the clones of T helper. Now how are they preparing? Let's see. First of all, the chromosome 3 has a gene which will produce the IL5R. In the same way like IL4R, the IL5R will travel towards the surface and get attached to the surface in order to wait for IL5 to come and fit into the IL5R on its surface. Now who is going to secrete the IL5? IL5 is going to be secreted by chromosome 5, a gene of chromosome 5, and actually by whom? The helper type 2 clone cells. This is the clone cell actually. This is the clone of T helper type 2. So again, a communication is established between the two clones, the two types of clones. As if in the same way their parents were communicating with each other, they are now starting to communicate with each other. Okay. Now there is a communication between next gen B lymphocyte clones and next gen T helper type 2 clones. And in the same way, the IL5, the golf ball, will fit into the golf pot, the IL5R. Now let us see what is the aftermaths occurring inside the B lymphocyte clone cell. The IL5 is actually acting as a stimulus for antibody production. Now which antibody? That antibody which was stimulated and was present on this clone parent B lymphocyte. So that antibody is going to be synthesized now by this stimulus. So getting the stimulus, immediately the nucleus gets changed into a cartwheel shaped nucleus. Now this cartwheel shaped nucleus is actually undergoing orchestration for the production of the antibodies. And during this process, after differentiation, the B lymphocyte clone cell can be called as the plasma cells. Plasma cell is the specific name given to the antibody producing B lymphocytes. Now see what's the fate of next generation B lymphocyte clones. Now they are destined to produce the antibodies. Now which genes are responsible for synthesis of the polypeptides present in an antibody? They are chromosome 15, chromosome 2 and chromosome 22. Now see, chromosome 14 produces, a certain gene of chromosome 14 produces the heavy chain of the antibody, that is immunoglobulin. Chromosome 2, a gene in chromosome 2 produces the kappa light chain of the immunoglobulin, and chromosome 22, a gene in it, produces the lambda light chain of immunoglobulin. Heavy chain is compulsory, it's constant. But the light chains are optional, any one may be present. And this antibody is going to become IgM or IgD, because already we have discussed that the surface antibodies present in the B lymphocytes were IgM and IgD, IgM or IgD, and the same will be produced. The clonally selected B lymphocyte, its descendants are going to produce the same antibody that was selected. That's why it is again the synthesized antibodies will be IgM and IgD. Now next, the antibodies come out, and thus the plasma cell is ready to produce the antibodies, and the human body says, ha ha ha, I am now ready to fight with you. It is talking with the pathogens, the bacteria, that I am ready to fight. I am ready to fight with you. And my weapon, my sword, is the antibody. Okay? Now, what's the benefit of antibodies? Antibodies are like jam on the bread. Bacteria, let it be considered as bread. And antibodies are like jam over it. Now, why am I using these two terms, bread and jam? Suppose you don't like bread without jam, but you like bread with jam. Similarly, when the bacteria is coated with layers of antibodies, the neutrophils want to taste it. They are interested in it. They are attracted towards it. So just in the starting of this video, I told you that the attack of bacteria or pathogens by the neutrophils is slow, but when it is covered with antibodies, it attracts a lot of neutrophils and rapid phagocytosis takes place. This is the advantage of antibodies. Thanks for now. We shall meet in next video. Until then, bye bye.